Hey folks, guess what this lesson is about? Yep, you guessed it, RNA. So RNA is really similar to DNA. We can call it like DNA's little sister or cousin or something like that. So the structure of RNA, let me see if I can move the screen down. There we go. Structure is pretty much the same as DNA. There's just one slight difference in the main structure, and that is right here. So the sugar in RNA is ribose as opposed to deoxyribose in DNA, and that just means that this regular ribose has an OH group right here. The deoxy, it doesn't have that oxygen there. So it doesn't seem like a very big difference, but that extra oxygen on this number 2 carbon here actually makes the molecule less stable. And so that's why RNA isn't normally the thing that organisms use to keep their instructions for their genes. We use DNA because it's more stable. And then RNA is just kind of used to make temporary copies. The other difference with the structure that you're going to see here is just with one of the bases. So in DNA, you have thymine, and this is the thymine right here, the nitrogenous base. And then in RNA, it uses a base called uracil instead. And those are very similar. Again, there's just like one little carbon group off to the side here that uracil doesn't have. So whenever you see a T in DNA or thymine, you're going to replace that with a U for RNA. So if you're doing complementary base pairing, then in RNA you're going to have a U base pairing with an A instead of a T. And then the number 3 thing is that in most organisms you're going to find RNA as a single-stranded molecule instead of a double-stranded molecule like you find for DNA. So here is, sorry, trying to go back. It's going to show you my faces in the way here. Here's a strand of RNA. So it polymerizes the same way DNA does. So you'll have this phosphate group, and then the ribose, and then the base. And then when it gets put together, the phosphate group of the next nucleotide gets attached on to that 3' end of the sugar of the base in front of it. So you get the same sort of phosphate, sugar, phosphate, sugar, phosphate, sugar backbone, and then the bases sticking out to the side. So we have U, G, C, and A. All right. Slide over here. So like I said, DNA is kind of the master plan for traits. DNA is the permanent copy of the instructions for our traits, and then RNA gets used to be sort of temporary copies of it. There's 3 main types of RNA. The one we usually talk about is mRNA, which stands for messenger RNA, and this is the little copy of genes, which are the instructions for building a protein, and this mRNA molecule here, it's in yellow, is basically a copy of those instructions that gets carried from DNA in the nucleus to ribosomes out in the cytoplasm. And there's a couple other types of RNA that we're not going to talk about too much today, but we'll get to them later. rRNA stands for ribosomal RNA. And so that's a molecule that just forms part of the structure of a ribosome. This is a ribosome right here. So it's made up of rRNA plus some proteins to make this big, funky protein building machine. And then the other kind is tRNA, which stands for transfer RNA. That's also involved in building proteins. So that's these little molecules here that bring amino acids to the ribosome and matches them to the instructions. The message is coded in the mRNA to build those proteins. So we will talk more about those in the next lecture today. We're going to be focusing on mRNA, which carries the messages of the instructions from DNA. And back to my corner over here. So today's big word is transcription. Transcription means basically the process of making a copy of something. You might hear the term transcribe. If you transcribe something, you're writing down a copy. Same thing. So in the transcription we're going to talk about, what happens is segments of DNA serve as templates to produce complementary RNA molecules. So DNA is like the model that's there, and then we're going to make some RNA that matches it, that's a copy of it. So in eukaryotes, this is going to happen in the nucleus because that's where all the DNA is, right? So there's a big enzyme called RNA polymerase, shown here, this big pink thing. It's a really cool enzyme, and it's going to come in and bind to DNA in the nucleus. And where it binds, it's going to separate the strands a little bit. And then it's going to use one strand, or read one strand of DNA, and use it to assemble a strand of mRNA that is complementary to it. So for instance, if it's coming along and it's reading this DNA and it sees a G, then it, on its mRNA strand, what's it going to put there? Well, it's going to put that complementary base that likes to interact with G, and that would be a C. So it's going to match up all the G's with C's, and it's going to match up all the T's with A's, and then it's going to match up all the A's with U's. Don't forget, RNA uses U instead of T. So it's just going to go along and read one of these strands and then add on mRNA nucleotides to the 3' end. Remember, everything gets built by sticking phosphates onto the 3' end. And it goes along and it makes this nice strand of RNA that's a nice copy of these permanent DNA instructions. So how does RNA polymerase know where to start? I mean, there's this big, long DNA molecule with tons of genes on it, and lots of space in between the genes, too. So RNA polymerase actually needs a little bit of help to know exactly where to start and stop. There's one spot that RNA polymerase is going to bind to on the DNA, and that's called a promoter. So kind of it promotes the gene. So in front of the gene is shown here in pink, right in front is this area called the promoter, and it has a special sequence of bases that shows RNA polymerase exactly where to start making the mRNA. And there will be regulating proteins that come in and bind to the DNA as well to help RNA polymerase find the promoter. Excuse me. And then, so RNA polymerase will bind here. It'll go through and it will read off this gene and make the mRNA. And when it gets to a spot at the end, there will be a special sequence in the bases again that's going to tell RNA polymerase to stop and let go, and it'll go its merry way and leave a strand of mRNA that's an exact copy of this section of DNA. And then you've got this nice section of what we call pre-mRNA. But these mRNA molecules need to be edited, so they're like the rough draft of an essay. They are going to have a protective cap and tail added to prevent the ends from breaking down. So here's the cap on the 5' end, and then there's a tail on the 3' end. And then also sometimes there's some sections that need to get cut out, so those will get spliced out. Then we have this nice mRNA. That's exactly what is needed, and it gets transported out of the nucleus, and it's going to go out to the cytoplasm to find some ribosomes, where it's going to provide the instructions for making some proteins. So that's my last slide here. After this, go ahead and watch the next video, which is a really nice 3D animation of what the process of transcription looks like, and then go ahead and start on your worksheet.